Hello friends, welcome to my channel. Today we are going to see about viruses. This is the very first topic in our PG TRB botany syllabus. So what is meant by virus? Virus is called as biological puzzle. Why do we call them as puzzle? Because it's an intermediate form between living and a non-living. So that is, it has both living character and a non-living character. What are the living characters? Now we'll see. So, virus has the genetic material. It may be either DNA or RNA, but never both. And it can reproduce its genetic material inside the living host cell. Next one is, it can mutate its genetic material. Example for this is common cold. The virus will be the same, but the strain will be different due to the mutation of its genetic material. And the next one is host specificity. That is, some virus can infect bacteria, some virus can infect plants, some virus can infect animals, and some may infect human beings. So, the virus which infect bacteria are called as bacteriophages. And the virus which can infect blue-green algae or cyanobacteria are called as cyanophages. And what about mycophages? Myco means fungi. So, the virus which can infect fungus is called as mycophages. And next one is, the virus has antigenic properties. That is, the virus has the ability to cause infection or to cause disease. This is called as antigenic property. And the next one is irritability. Irritability means response to stimuli. That is, the virus can respond to ultraviolet radiation, chemicals, radiation, etc. And now we'll see about the non-living characters of virus. That is, the virus is inert outside the living host cell. That is, it requires living host cell to replicate its own genetic material, and also for protein synthesis it requires living host cell. So next one is, just like non-living material, the virus can be crystallized. So the first virus which was crystallized by W.M. Stanley was tobacco mosaic virus, and he crystallized in 1936, and he got Nobel Prize for this in 1946 in the field of chemistry along with Dr. Northrop. And next one is, there is no functional autonomy. That is, the virus cannot replicate its own genetic material, and it cannot produce protein synthesis without help of living host cell. And they have no protoplasm, and they have no metabolism, and there is no energy production also. So these are the living and non-living characters of virus. And next we will see about size. The size may be from 20 to 300 nanometer, and the bacteriophages size will be 10 to 100 nanometer. And based on the arrangement and the number of capsomeres, there will be three shapes. First one is cubic symmetry, adenovirus, and 5x174. These are the examples for cubic symmetry, and tobacco mosaic virus is the best example for helical symmetry, and the bacteriophages are best example for complex symmetry. Complex means both forms will be there. Cubic and helical forms will be there in a single phage. And next one is classification. Mainly the viruses are classified based on nature of host. That is, whether the virus is infecting plants or viruses. The virus which infect plants are called as plant viruses, and the virus which infect animals are called as animal viruses. And next, Baltimore has classified viruses into 7 classes based on the nature of genome and DNA or RNA, use of reverse transcription. RT means reverse transcription, and positive or negative RNA. So these are the criteria Baltimore has used to classify the viruses into 7 classes. First one is nature of genome, which means whether the genetic material is single-stranded or double-stranded. And the second point is DNA or RNA, whether the genetic material is DNA or RNA. And the next one is use of reverse transcription. That is, whether the virus is using reverse transcription to replicate its genetic material or not. So what is meant by reverse transcription? Reverse transcription. This is also called as teminism. This was discovered by Baltimore and Temin. So before we know about reverse transcription, we should know the central dogma of the cell. Central dogma of the cell. What is meant by central dogma means each living cell have DNA. This DNA is transcribed into mRNA. Then this mRNA is translated into protein. This is the central dogma of the cell. This is called transcription. This is called translation. Conversion of DNA into mRNA is called transcription. Conversion of mRNA into protein is called translation. So here we call it as reverse transcription, which means the virus has RNA. This can convert reversely into DNA. This DNA is called complementary DNA or cDNA. So the virus mRNA is get converted into DNA or cDNA, complementary DNA, and this cDNA again produce RNA and then proteins. So this is called the reverse transcription. So he used this criteria to classify viruses. And next one is, fourth point is positive or negative RNA. What is meant by positive or negative strand? Positive strand means this mRNA, now we have seen mRNA is used to produce DNA, and then this complementary DNA produce RNA and protein. But in case of positive strand, positive single strand RNA, here positive means this RNA can directly act as mRNA and produce proteins. That is, this mRNA can directly act and produce proteins. There is no need to produce cDNA. Okay, so this is the meaning of positive single strand mRNA. So what are the 7 classes means? First one is virus with double strand DNA. Example for this is adenovirus. And virus with positive single strand DNA, that is parvovirus. And the virus with double strand RNA is reovirus. And the virus with positive single strand RNA is toga virus. Virus with negative single strand RNA is rhabdovirus. Virus with positive single strand RNA with reverse transcription. So this reverse transcription occurs in retroviruses. Here DNA will be intermediate. And next one is virus with double strand DNA. Here also reverse transcription is there, but here RNA is intermediate. Example one, the hepadnavirus. So next we are going to see about the structure of virus. Actually virus has 2 parts. One is protein and the next one is nucleic acid. This protein united to form capsid. So capsid is just like protein coat, and capsid is made up of smaller units, smaller subunits, which are called as capsomeres. The nucleic acid may be either DNA or RNA, and some proteins have envelope. So this is made up of lipoprotein, and the enveloped virus are called as lipovirus. So how the virus get this envelope means it is derived from the host cell plasma membrane during infection by budding. And next one is the envelope. Outside the envelope there will be projections. On the surface of the envelope there will be projections. These are called as spikes or peplomers. Actually they are helpful for the interaction with the host, and also it fuse with cell membrane of the host for the infection. So these are the functions of spikes or peplomers. So we see example for virus is tobacco mosaic virus. This is a helical rod shaped virus, and it is again made up of capsid and a single stranded RNA. Capsid is made up of 2130 capsomeres, and each capsomere is made up of 158 amino acid. Here helical rod shaped means this is the helical shape. Okay. So next one is the nucleic acid and protein content of TMB will be, nucleic acid is 5.6% and protein will be 94.4%. So this was the first virus which was crystallized by Stanley in 1935, and as I already told, he got Nobel Prize in the field of chemistry for crystallization of TMB. And this virus caused tobacco mosaic disease in tobacco. So next we will see about bacteriophages. What is meant by bacteriophage? The virus which can infect bacteria are called as bacteriophages. So the bacteriophage was first discovered by Twert. Okay, and the term bacteriophage was coined by D. Herrell. So the virus which can infect E. coli are called as T phage. So there are variations T1 to 7 which was enumerated by Delbruck. Okay, next one is the bacteriophage has head and tail. Okay, head is hexagonal shape and tail is cylindrical in shape. Here the nucleic acid will be double-stranded DNA, and there are three parts: head, tail and collar. Collar is uniting the tail with head, and there is a base plate. Okay, which this base plate which secretes the enzyme lysozyme. This lysozyme helps in penetration of the virus genetic material into the host cell. And next, this phage base plate has 6 tail pins or pegs and also 6 fibers. Okay, so that there is a difference between lambda phage and bacteriophage. Lambda phage has single pair of fibers, whereas the bacteriophages has 6 fibers. Okay, 6 tail fibers. So next we will see about life cycle of the virus. In bacteriophages have life cycle which are of 2 types. First one is lytic and next one is lysogenic. What is meant by lytic cycle means the virus, that is the bacteriophage, can infect bacteria and reproduce inside its cell, and it can lyse or kill the host cell, that is the bacterial cell, and release in large numbers. This is called a lytic cycle. So there are 5 steps in lytic cycle. First one is adsorption, next one is penetration, and next one is synthesis, assembly and maturation, and last one is release. Adsorption means the bacteriophage recognize the host bacterial cell and will absorb on the host surface, that is the bacteria surface, and this recognition is called landing. So first step is landing. Here the phage, that is the virus, recognize the bacterial cell. This is called as landing. After recognition, the base plate, that is the fibers and the base plate will attach on the host surface. This is called pinning. Okay, this is the second step of adsorption. Next one is penetration. After absorption, the contractile sheath which surrounds the tail of the virus, of the bacteriophage, contract and penetrate its genetic material into the host cell. Here the penetration occurs due to the presence of lysozyme enzyme which is produced by the base plate of the bacteriophage. And after penetration, the nucleic acid, that is the genetic material, will go inside, and the capsid, that is protein coat, will be there in the outside. This protein coat is called as ghost. Okay. So next one is, third point is synthesis. After entering into the host cell, it takes the control of bacterial, that is host machinery, biological machinery. It takes the control, and takes the host machinery, biological machinery, and it takes the control and command the host genetic material to produce viral genetic material and viral proteins. Okay, so this is called synthesis. Next one is assembly and maturation. So here the head particle, that is the protein part, and nucleic acid tail will be produced separately. So all these particles, head, tail, and collar, all these particles, protein particles, will be assembled. This process is called maturation. So after maturation, the host cell wall will be lysed, and the bacteriophages will come out in enormous amount. For example, after 20 minutes of infection, there will be 300 phages will be produced. Okay, so this is all about lytic cycle. So this is the diagrammatic representation of lytic cycle. First, the bacteriophage recognize and attach on the bacterial cell, and next one is here the genetic material is pierced into the host cell. Next here third step is the protein coat will be outside, this is called as ghost, and the synthesis of virus own genetic material and proteins. And the fourth step is assembly and maturation. Here the proteins and head particles all will be assembled. And last stage is release, that is the cell wall of the host cell will be hydrolyzed by the lysozyme enzyme, and the phage particles will be released. Again this phage particle will infect new host cell. This is diagrammatic representation of lytic cycle. Next cycle is lysogenic cycle. Here the bacteriophage will recognize and adhere on the host surface, and in lysogenic cycle the DNA, that is the genetic material, is pierced into the host cell, and it does not will free in the host cell, that is it integrate with the host. This is the bacterial DNA, and this is the bacteriophage DNA. That is the virus DNA is integrated with the host chromosome, host DNA. So after exposure of UV radiation, it can lead to lytic cycle, and the bacteriophages will be released. So this is lysogenic cycle. Here the integrated chromosome, that is integrated viral genome with the bacterial chromosome, is called prophage. This is an important MCQ, so please note it down. So now we have seen just the intro and structure of viruses. In next class we will see about diseases which are caused by virus in plants. Okay, and we will also see about virion, what is meant by virion, what is meant by viroid, and what is meant by virusoids, and what is meant by prion and interference. In next video we will see about all these things, and I will give you few questions which are important for our exam. First one is, what is meant by mosaic cran height, and next one is, who described lysogenic conversion, and the third one is, who described ultrastructure of TMV, that is tobacco mosaic virus, and fourth one is, what was the first cyanophage discovered, and the fifth one is, who was the first to culture a plant virus, and the sixth one is, what was the first enzyme reported from influenza virus. So please try to find out all these answers for these questions, and we will discuss in next video. Thank you.